Welcome to our data science meeting dedicated to FAIR principles with Alise Ponsero from Core Bioinformatics.
Yeah, so thank you for being here today. So basically, this session is going to be part of two sessions. So talking about FAIR principle, FAIR data principle in particular, and kind of building together a reflection around what we want to maybe improve in terms of data management at QIB, and where are your pain points when it comes to data. So this session is going to be mostly around omics, so genomics and metagenomics, because that's what I know. And the next session in July is going to be from BioFair, and they're going to talk more generally about fair data principle in biology in general and what they are trying to build with the BioFair initiative. So you probably all have heard about the fair data principle. So those principles were published now almost 10 years ago. And I would say that in such a short time, it's quite striking that there have been like a really growing recognition about the importance of data management, and more importantly, a real push from institutions and funders to promote fair data principles in all sciences. Fortunately for us, there have been also a lot of tools and methods and standards that we can apply to make our biological data fair. Because those fair principles were developed kind of with like all data in mind, and there are specificity of omics data that may be a bit more difficult to navigate. So today I'm going to mostly, when I talk about data, I'm mostly referring to sequence data and the specificity of making that type of data set a bit more fair. So the first two principles in the FAIR acronym are findable and accessible. Those two are typically the easiest. And that's usually the one that we easily can meet with limited effort. So the findability means that your data is associated to a unique identifier that is persistent in time. So if you're talking about sequence data, it will be the SRA ID that you have for your sequence data. And the findability also requires you to have the data indexed in the repositories. So it means that people can search and find it, basically. And the accessibility is just people can search and find it, but they also can access it. So they can download it for free. That is definitely important. And that it's using an open source protocol. Authentication is totally possible in the FAIR principle. It's just only applied when necessary, so for sensitive data or things like that. So basically, when we submit our sequence data to a sequence archive, such as SRA or ANA, or if you're talking about more generally other type of data, you can use generalist archives such as Zenodo or Figshare. And basically, you will meet those two requirements quite easily. You will have a unique identifier, it will be indexed, and people can download it and access it. So more and more, when you submit your study for publication, there is a push from the journals to have a data availability section at the end of your paper, where you need to link the unique identifier to your study. So basically just citing the name of the archive where you deposited the sequence, the project ID, but if possible also the identifier of each individual sequence that you've used should be linked to your study. So it's quite straightforward, even if Sumit would disagree that submitting sequences to SRA is straightforward. But despite this, today we estimate that a fifth of all metagenomic papers don't have the basic findability and accessibility of metagenomes. Most of them do not have an availability section, and that explains it. And some, it's probably a typo, the identifier is not correct or it doesn't exist anymore. But I would say that we are getting better as a field on those two aspects in particular. Things are definitely evolving fast, and it's really getting better. The two other points, interoperability and reusability, are way more difficult. And that's usually where the pain point is when we talk about fair data for omics datasets. So reusability and interoperability kind of means that people can reuse your dataset and understand it and use it in conjunction with other datasets. And that requires you to basically provide some information about the sequence itself, like where the sequence was coming from, what was the isolates, how did you collect it, when, etc. So you basically need to submit your sequence along with a contextual metadata, and those two components need to be found and accessed. This contextual metadata also needs to be using things like controlled vocabularies or ontologies, if possible, so that people can actually reuse it. So as I said, this is definitely the most difficult part, and you probably all have searched in ENA and tried to look at the metadata associated to a sequence, and you probably know that it is quite an experience. And this is true for a lot of very important data sets. There is this commentary paper that I really like from the University of Arizona, where they showed that for the 2 million COVID viral genomes that we have generated during the pandemic, only 33% of those genomes had any basic contextual metadata, like where the isolate was coming from and just where and when. That is only available for 30% of the COVID genomes. And what they argue in that commentary paper is that if we want to be able to have any kind of international collaborative effort like we had in the last pandemic, we absolutely need to address that. And they are quite bleak in the commentary. They basically say that to prepare for the next outbreak, we need to address it. And I totally agree. I think this is definitely the priority when it comes to data management. So when you submit a sequence to SRA or ENA, you probably have come across those types of lists. Those are the mixes standards. So in order to make your dataset a bit more reusable, the repositories will ask you to submit that minimum metadata in order to understand the sequence. So here you have the list. It's not easy to read, but you have the list for the human gut metagenomes. And you basically see that it's a title for your sample, a collection date, latitude and longitude, and a bit of description, like if it's a fecal metagenome or if it's something else. But that's all. That's the minimum required value in order to submit your metagenome. And what is even more difficult is that very few of those fields have any type of data check. You can see that a lot of them are text fields. So if you make a typo, well, people cannot reuse your metadata. And ENA is pretty good because at least the latitude and longitude and the dates is a regular expression, so it's checked, but SRA, it's not the case. There have been some efforts to help people submit the right minimum metadata associated to the sequence, but it's really recent and it's a bit difficult to use still. The other difficulty when you submit your data to those types of repository is that the data model is really meant to accommodate most studies. So like here you see the data model that is used in ENA. And you see that basically you are expected to give some information about your study, so a title and a goal. you are supposed to give some information about the sample that you collected. So that's the raw biomaterial. And then you are supposed to give information about the experiments, so the library preparation and the raw reads. And you see that this is really a nice data model for most cases, but if you have things like a longitudinal data set or a column model fermentation, this is quite challenging to because those samples are not independent from each other. So it can be challenging to keep that information in that framework. The other thing that is not possible with that framework is that if you have non-sequence measurements that were done on the same sample, it's quite difficult to link it. Let's say you have done a metabolome on the same sample as your metagenome. Well, you cannot deposit the metabolome data in SRA. So you would need to somehow link it to another repository or a supplementary material in your paper. And finally, there is no easy way to submit non-sample sequences such as blanks, because by definition, a blank is not a sample. So how do you deposit your blank in that model? So it's not perfect, but it's kind of challenging to make things understandable in that framework. And to illustrate that, this is an example of a study where they have an infant cohort from Danish babies. And if you read the paper and the supplementary material, you are able to understand that those two samples are coming from the same infant, the infant number 10, and this is taken at 1 year and this is taken at birth. But if you don't read the paper, this is not apparent. It's just the sample name is 10_12M and the other is 10B. So yeah, this is not super clear. It's not even clear that it's from infants and what is the age. So globally, I would say that, at least in my opinion, the main difficulties when we talk about fair data in omics is interoperability and reusability. And this is because today our standards are not enabling us to capture the complexity of what we are dealing with. And so the question is not really to replace those standards, but kind of enriching them and finding ways to fit better the institute needs or your needs in particular. So in this presentation, I'm going to mostly talk about two research experiences that I have done previously from those two beautiful labs to kind of discuss around what is possible to do when it comes to fair data in different settings, and maybe like brainstorm about what you would need to implement for your own research. And the first example is a work that I've done in marine microbiology. So before moving into gut microbiome, I was a marine ecologist, as those pictures of me trying not to be seasick can attest. And why fair data in marine microbiology is super important is that when we look at the marine microbiome, it's a bit like the gut microbiome. You cannot understand it if you don't have the global ecological contexts. Because marine microbes are super important to understand food webs and global elemental cycles. So you kind of need to understand what is the composition of the seawater, what is the temperature, what is like the setting in which those microbes are living in order to understand your microbial community. And that's where having good data sets is absolutely necessary, especially since we are talking about planetary scale analysis. Fortunately for marine microbiologists, there have been a lot of great international efforts to sample the ocean. You probably have heard about Tara Oceans and OSD. Those are like large-scale international collaborations to sample most seas and oceans. And then we have also beautiful time series such as HOT and BATS, which were sampling the same location for over 30 years. So those are great data sets, and it represents terabytes of sequence data all around the world. And of course, those beautiful data sets are not interoperable with each other. They don't use the same vocabulary. They use all different units, which is not nice. And they all use different type of way to measure those data. An added difficulty when we talk about marine datasets is that the physiochemical measurements that we do on the seawater is usually deposited in a different type of repository. So it will be Pangea or Bicodemo, whereas your sequence data will go to SRA and ENA. So we kind of split always kind of the data sets into when we submit the data. So my first postdoc was about trying to create a web platform to reintegrate those data sets together for those historic and high-value data sets. So the main goal were to first harmonize the different sampling expeditions together, to reconnect the different components, and then hopefully have a nice database where people can use the data sets. And I would say that the most difficult part of that project was the first step, which was the semantic harmonization. And what I mean by that is that we would have those metadata table coming from the different cohorts, and you see that for Tara Ocean we would have a column called nitrates, and in bats we would have NO3. If you have kind of read a bit the literature, you can kind of guess that it's talking about the same type of measure, but I mean probably the unit is not the same because like the scale is very different. So it means that basically you need to read every paper and go back to the original protocol in order to make sure that what we are talking about in this column is actually the same across the different data sets. So we did that, and in order to capture this, we basically have added a JSON file describing the metadata. So it's kind of a metadata of a metadata. And we call that a semantic layer. It's basically just a document that will capture all that we know about this metadata. And in that project, we decided that there was 3 elements that we absolutely needed to capture for all the variables, which was what the variable was about. So this would be the concentration of nitrate in liquid water, as an example. What is the unit that we are using? So that would be micromole per kilogram. And then what was the measurement? How was the measurement done? So spectrophotometer, as an example. So we basically did that for all the variables in all the projects, and it literally took us a year or 2. So it's a huge effort to manually have to do that. But once it's done, we can then reconnect the different pieces. The next step was to try to build a data model that would be fit for marine metagenomes. Because you cannot really understand the sequence file that you have if you don't have information about the sample, so the actual water taken at the same depth. But you also need information about the column of water. So you may need to reconnect your microbiome data to the microbiome data taken 5 meters above or 10 meters below. And you may need to look at the gradient of temperature. So you need kind of the information about the column of water and more globally about the oceanographic movements. So you definitely need to reconnect the local scale to the bigger scale in your data model. So we tried to do that. We basically went through all the repositories and asking for colleagues to send us metadata. In order to collect from information about all those components in order to build a database that would reconnect those different components together. We would slap the semantic layer on top of that in order to build what we call a data package. So a data package is kind of this idea in FAIR data that you have a self-contained data set and if you send that data package to anyone, they have all the information that you actually need to reuse it and understand it. In that project, we were using the frictionless data standard, which is a toolkit that allows easily to package those types of complex data along with semantic information. And it's a nice self-contained standard. At that point, we decided also to store those frictionless data package in GitHub and Zenodo in order to save it for posterity. But we also used it to feed into our database. So there was kind of two outputs in that project. First, the data package itself, and then the database, which is building upon those packages. So yeah, in the end, we managed to get it up and running. And it basically enables people to search more generally on those historical datasets, both on the location, but most importantly on the semantic. So you can search for datasets that have a concentration of nitrate in water between 0 and 10, as an example. And yeah. I would say that the most important part is that it enabled people to do meta-analysis on those datasets. So now that the datasets are pretty and interoperable, you can actually use them in together. Here you have a paper where we looked at the distribution of some taxa across different physiochemical gradients. Here it's just depth, and those are pretty plots, so I like them. So yeah, so the main take-home message about this first example is that there is this concept of having a self-explanatory data package that you can have, where you basically have your sequence data, so your actual data, you have a metadata about you, explaining your sequence, and you have a semantic layer that describes the contextual metadata. And that altogether kind of is self-contained. I would say that it's having done that effort, it's a lot of effort. It's a lot of time. In total, that project was 3 years full-time for 4 people. And it's not that many datasets. So it's a time investment, and but I would say that it's definitely valuable for high-value datasets that you think could be reused long-term. I would say that now that I've seen the gut microbiome world, I understand also that not, contrary to marine metagenome world, not every data should be publicly available. We worked with sensitive data, and if you're currently working on your dataset, you may not want to make everything publicly available right now. But I would argue that it's still good to kind of plan for your FAIR data, to apply FAIR data to your datasets, even if it's not completely publicly available. And I'm going to explain why. So my previous postdoc was in Finland on a cohort of more than 1,000 families, and it's a beautiful data set where they sampled from birth to now 5 years old the infant and their parents at different age. They collected also milk samples, dust sample, a lot of samples were collected. And most importantly, there was an extensive amount of questionnaires that were asked to the parents. So all the red lines are the regular questionnaires. So it was basically every week the parents would have to answer questions about their babies. And then there was like growth questionnaires and things like that. So in total, it's a huge amount of metadata that was collected for that cohort. And when I joined their lab, they were at the point where they finished the 2 years collection, and they were planning for the 5-year follow-up. And it kind of became very apparent that we needed absolutely, in order to make that dataset usable, to have some type of curation and harmonization of the questionnaire data. We needed also ways to track the metagenomes and the data that we were producing. And we also needed to take into account that we were working with international people, all looking at different angles at these datasets. And we also needed a way to track issues and contamination and things like that. So we decided to create an internal database to kind of follow all the outputs and all the data that we have about that cohort. So we had the questionnaires. Briefly, like this is the model that we followed. We have the families, the questionnaire as to the families, all the samples that were collected for that family. And the first task that I had was to create a controlled vocabulary, meaning that we needed to describe accurately what variables were measured, in particular since the original questionnaire were in Finnish because the people were, the families are Finnish, and we are an international team, so we needed English translations. And we needed to agree on what we were talking to kind of have a common language to learn to use that dataset together. We also added in the database an easy way to track if any samples had or run had an issue, in order to make sure that we didn't use datasets that were, contaminated or things like that. And we added a way to add in the database which people were working with, which subsets of the project and which outputs were produced, to kind of track everything that was going on with these cohorts. So what I mean by that is that we had this modular database, internal database, that allowed us to work together internally, and that we could connect to the public as we go, to connect it to the publicly available side of the datasets. And I would argue that this is definitely valuable, even if the whole dataset is never completely publicly made available, to have an internal fair dataset, because it will allow you to first reuse it long term, or make sure that when the person who made the dataset leaves the lab, that a follow-up is possible, and that you can use it in conjunction with other datasets. So I think it was 2 years ago in the project, we got contacted by some people in Norway, and they wanted to do a meta-analysis of all the infant cohorts in the world, and they wanted to add the Helmi datasets in their ARG analysis. So having that database internally really allowed us to easily kind of give them the subsets of information that they wanted and that they needed, and reconnect it to the other cohorts. So it can be, I think it is valuable even if it's only internally. So kind of a summary of what I just said is that you can totally make your part of your data publicly available and fair, while retaining the sensitive contextual data internally, and making sure that it's still available long term. So it will be upon request. Some researchers may ask for that sensitive data to be communicated to them, but it needs to be there for a long time in your hand. So what I've discussed so far is mostly that we can think of ways to apply the FAIR data standards at any point in your data journey. So before publication, I would argue that having in place an internal data management process definitely is valuable if you don't want to lose data or work with other collaborators, or if you have in your hand a very complex data set, such as the motion data set. then you may want to have something that's a data object internally that you can interact with and that is kind of fair. At publication, parts or completely the data object can be made available. And at that point, the data package becomes kind of fixed, a fixed entity that will not move. And then long term, I would say that having a series of nicely curated and completely fair data objects is super valuable if you're planning to have, here it would be cohort 1, 2, 3, but it's fine. If you need to bring together disparate data sets and use them in a meta-analysis, or if you want to do a cross-study analysis. So, where I'm kind of interested in talking with you is maybe kind of reflecting of what are your current pain points when it comes to data, managing your data in your lab. Knowing that there will be a data science meeting in October talking about drawing the data management plan, like for grant submission and things like that. But if we focus only on once you have your data generated, do you think that you have any? What is your experience in managing the data? We have all used Irida, but is that enough? We know that the metadata associated in Irida is kind of not always complete, I would say. And we were wondering if that is something that's, when you ask to put in Irida, why the metadata is not always a priority? Is there anything in the way we are currently managing the sequence data that you think is difficult for your projects? Can you retrieve, as an example, datasets from your previous students in your lab easily? Do you have kind of have you ever tried to retrieve datasets from someone else in your lab? Or even if once at publication, like if you have any, we know that people have been. We have a way to submit for you to SRA, but sometimes like the metadata associated to it is a bit difficult to understand because like we don't know the dataset as you do. So we can follow the MixS standards, but sometimes it's not always the most relevant way. There was also I had also some question, like if you have other types of outputs than sequence data, like maybe some of you have generated like protein catalogs or things like that are not the classic sequence data. And those are difficult because we cannot submit them to SRA. So you would need to submit them to something else. So I don't know if you generated those types of data. What do you do currently in the institute? Yes.
So usually I generate metabolic models. So I do generate metabolic models, and we do have EBI database called biomodels. They do have some standards that needs to be followed, and they do their automatic checks. But having said that and having done that, the models in biomodels are not reusable mostly because, there is always something different or the SPML file. So I think it's, even in the modeling field, it's a problem that even though we have a way or a standard to submit it, is ultimately not reusable by the community.
And is it because like some part of the information is missing, kind of
Yes.
like for the
Yeah,
metagenomes?
yes. So more like metadata, but within SPML you can have different approach how it is generated or how the model was generated, so that information is not there. I'm not sure if I'm answering your question. I'm just adding more. I'm just complaining that, yeah, it
That's what
is.
we are for.
Yeah, listen to my complaints. Yeah, so it's a big problem, and it would be good that the community comes, like Biofair is a good community approach that they are taking and being funded to get it more reusable within the community. And metabolic modeling community is very small, and still we can't use each other's models. So that's a big problem, I think. Sorry, that was my complaint.
No, this is definitely good to, yeah, that, because like I feel like the metabolic models are kind of, it feels like it should be less messy than metagenomic data, because yeah. So I'm kind of surprised, but not that surprised.
Similar problem what you were showing with nitrate and NO3, even with metabolic models, is the metabolized naming. One model will use nitrate, another will use NO3. You can compare because they are different identifiers. And they exist at every layer, not just at metabolite, at reaction level, at every layer. It's how do we make it consistent identifiers within a big database?
Yeah, and that's where, like in the example that I showed, is like that's why we decided not to rename everything consistently, but just rather adding kind of a description of the variable, because everyone has their own ways of encoding a variable. I think the main thing is like to keep the information, having either a controlled vocabulary or the best would be an ontology term, in order to describe accurately the. But in the case of a metabolite, like there are some ontologies for metabolites, right, that could be linked directly.
So there are like many biochemical databases that are used to generate these models, and every database has their own identifier. So I mean, like similar to what you did, a big effort to map identifier between databases of Cohort. There was a German group who were trying to do and map all the databases metabolic so that you can identify if it's nitrate in one database or is in another database. I don't know where it went, but yeah, there was similar effort, but I'm not sure if it was completed.
Yeah, and it's kind of a race, like against time, like to curate those things, like because as people publish, then you need to constantly like curate and add, and it would be better if it was clean from the beginning.
Is there any kind of like, I mean, I can imagine it's be quite complex, but any kind of platform that either already exists or could be created where it's got like the code or algorithms written in, so when something's uploaded, obviously it would have to be continuously updated or things added to it data, but for example, like anytime any of these things are picked up in this data set, we name it to this, just to like, for example, like the nitrate thing or something like that, have like a series of terms that are commonly used, and then just point it to like a single output, so like please change this.
Yeah, definitely there are some type of, there are some efforts to try to have like some kind of validation indeed of the fields, but that is really possible only for the like kind of the simple one. Like as an example, for the baby cohort, like if you report birth mode, and it's a simple one, birth mode, but some people will report only like it's C-section or vaginal delivery, but some people will say like, oh, but there was like antibiotic, intrapartum antibiotic. So like there are different, even for very simple measurements, there are different ways to encode it.
Yeah, not like binary.
Yeah.
It needs an exception on everything.
Yeah. So I mean, for marine and like anything that is like more not questionnaire based, but like real measurements, it's a bit easier because you can say like this is the concentration of nitrate in seawater and it's in micromolar. But for like questions, it depends also, like if it's questionnaire data, it depends how the question was asked to the person, and that makes it even more complicated, I would say, to standardize. But yeah, I agree. It would need, there definitely is a need, at least for the simple one, to have a bit more validation. Especially for the mixes terms that can be validated. As an example, in the mixes list, there are it's the broad scale environmental context, local environmental context, and environmental medium. Those three are supposed to be ontology terms from the ENVO ontology. And I really don't understand why it's a text field, because like it should be the ontology term ID, and you can validate that. But right now it's a text field and people can make a typo because like ontology terms kind of are difficult, and or would not understand that it is supposed to be an ontology term. So yeah, it's not great. It's not.